Today, I'd like to talk to you about bioengineering myths that everyone believes. And as I am a plasma bioengineer, a lot of this will be biased towards my background, but hopefully it will be at least a little bit useful for you. I'll talk about common misconceptions in bioengineering, where oversimplification could cause some problems, electric discharges, cells, and biological systems and how they interact, and separating mechanism from marketing. Hello, my name is Greg Friedman. If you enjoy serious discussions about science without hype or oversimplification, well, this channel is for you. Like, subscribe, and leave a comment. It really helps the channel grow. And this being very early stages of this channel, you can really influence where I head with it by asking me questions in the comments section. That's how I come up with most of these video ideas. Well, anyway, again, my name is Greg Friedman. I have a PhD in bioengineering. I'm former faculty at university for over a decade. And now I run my own plasma bioengineering company for almost a decade. We focus on atmospheric pressure, non-equilibrium plasma discharges, and their applications in medicine, environmental remediation, and the like. So let's talk about bioengineering myths. One of them is more power, more effect. That is certainly rarely true in biological systems. Those are highly nonlinear. Dose response matters. The dose response curves very rarely are a straight line. The example I like to give is if you take your house, I can come to your house at 5 in the morning, go to your bedroom, and knock on the door very quietly and say, hey, time to get up. You're going to miss the train. Or I can sneak in with a bucket of ice cold water and dump it on your head. Now, it's the same me, but the response from you is quite different. So what treatment was delivered, how it was delivered, and the dose and dose rate at which it was delivered matters a lot. Too much energy causes damage. I talked in a previous video about difference between dose and dose rate, and briefly, is gallon of whiskey a lot? Well, it depends. Gallon of whiskey in one evening is terribly too much, but gallon of whiskey over the course of 1 year, taken at a half a teaspoon in the morning and a half a teaspoon in the evening, has a very, very different effect, you would agree. So dose and dose rate matter, especially at the intersection of biological and engineered systems, which is what bioengineering is. So optimization is what's key, not just more power. Another myth is in vitro and in vivo, just about the same thing. Couldn't be further from the truth. Cell culture is not the whole mechanism. And I often read papers about different research related to cancer treatments, less so these days. But I remember back in grad school, my joke was that soon they're going to come up with a paper that salt kills cancer. And lo and behold, one day I stumble on a paper of anti-tumor effects of sodium chloride. Salt kills cancer. Yeah, you take a bunch of cells, put them in a petri dish, you put a bunch of salt on top, all the cells are going to pop from osmotic pressure, and there you have it. Salt kills cancer. You know, if that leads to a bunch of people running to the grocery store, buying pounds of salt and eating it, in vitro and in vivo, not the same thing. There's no immune system in a petri dish. There is no vascularization. There's no metabolism. Petri dish isn't the whole organism. Transition from a petri dish is very non-trivial. Petri dish experiments are important. They kind of give us kind of direction. So back to the stupid example of salt kills cancer. Okay, so now let's move on to a bigger model, maybe an organ on a chip model. Let's feed that organ on a chip high saline solution, like high salt concentration solution, and see what happens. That would be my next step before moving on to something like an animal trial where effects could be quite different than originally expected. Another myth in kind of the same direction is reactive species are all bad. Well, I mean, you drink antioxidant tea because oxidants are bad. Not always, not necessarily. Another kind of a joke I do with especially undergraduate students or high schoolers, it's a trick question. How much free oxygen is inside of your body right now? I mean, you're a machine that runs on oxygen, right? So the question is, how much free oxygen is inside of you? The right answer is none. All of the oxygen, it dissolves in the lungs and reacts with hemoglobin immediately and is transported through your body very carefully, delivered where it's needed and handed off from molecule to molecule. So there is no free oxygen. When you open up the body, like maybe have a paper cut, oxygen triggers extrinsic and intrinsic blood coagulation cascades, very important processes for blood coagulation, triggers wound healing, revascularizations, angiogenesis, and a host of other processes. So reactive species speed that up, and quite significantly in certain cases. I just learned today, rubber dandelions that are used to produce natural rubber, they produce natural rubber in the root. And they don't do it so well in a perfect hydroponic system. The roots need certain level of stress to maximize rubber production. Well, so does your body. To heal a wound, you need a certain level of stimulation, a certain level of stress is quite helpful. One example is your immune system uses a combination of nitric oxide, peroxynitrite, and superoxide, an ion O2 minus, to heal wounds and to fight infection. And those can be considered reactive species. They're not as reactive as their twins produced in plasma, but they're still reactive species. And controlled oxidative processes, they could be, and frequently are, quite therapeutic in the whole body. So the context determines the outcome. Another myth is mechanism is obvious. Correlation is not always equal causation. Multiple pathways, especially when you're dealing with a super complex evolved mechanism like a human. A lot of things in a human body are not perfect. They happen to have evolved that way for multiple reasons that this video is just way too short to get into. But your body frequently has many different ways that it can achieve the same result. So mechanisms are rarely obvious, especially in bioengineering. You're introducing two complex fields together. So on one hand, you have biology that is complex and complicated. And on another hand, you have engineering that is also, it has its own complexities. And now you're mixing them together, only further increasing complexity. So nothing is ever truly obvious. There is surface chemistry versus bulk chemistry. Like you inject a drug intravenously, you take a pill, or you deliver treatment or a drug directly to the target. Those are way different approaches to treatment, and they have different mechanisms and frequently lead to different results. So for this reason, mechanism isn't obvious. So mechanistic rigor is necessary and challenging. Another myth, if it works once, it scales. Nah, this isn't physics. In physics, you can do experiment once. As long as it's carefully controlled, you take a brick, put it on a table, and keep pushing it until it reaches the edge. You push it off the edge, and a brick falls. There. You tested the gravity, and it still works. You don't need to repeat that. Physics is simple, very simple physics. Like in my example, is if it works once, it's gonna work. In biology, because of the complexity of mechanisms, you thought the mechanism is simple, you tested it, and bam, it works. But it could have worked because of the phase of the moon. Don't discount weird things that science can't even explain. There is environment and sensitivity to the environment. And, maybe the patient was really, 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 really sad. That, I don't understand why, but that often affects wound healing. And also, there is variability because I'm talking about bioengineering. In bioengineering, you have some kind of device interfacing with some kind of biology. And no device is truly equal to another device. No matter how much we try to make the same exact copy of a device, there are tolerances. Plus minus a percent, plus minus a few percent, and that could have potentially devastating effects. So there's variation that needs to be accounted for, and in bioengineering, you have to test many, many, many times. If it worked once, does not mean it's going to work again. So there are many, many scaling challenges. Now, speaking about my field, a lot of times my colleagues mention, plasma is just oxidation. You're just oxidizing the body. Yeah, kind of. Non-equilibrium discharges are called non-equilibrium for a reason. There's a lot more stuff going on than just oxidative stress. There are non-oxidative reactive species. There are solvated electrons. There are multiple reductive species that are present. There are many electron-driven pathways or charge catalysis of biochemical reactions. So the complexity is far beyond simple oxidation. And I'll leave it there. Another myth I hear often is biological systems are simple targets. Oh no. Cells adapt, for one, right? They are in and of themselves, even though they're part of the whole organisms, they have mechanisms by which they can adapt to whatever treatment they're receiving. There is a feedback loop for both individual cell and whole organism and combination of cells and organs. Gene expression can change and frequently does change over time. My all-time favorite biological organism is so-called Deinococcus radiodurans. It's really cool. You gotta look it up. If you're into UFOs, Deinococcus is like the only proof I know of, true proof of alien life forms on Earth. Like this guy, it's like 4 organisms, 4 bacteria hanging out together, kind of holding hands. They have 8 individual copies, 8 different copies of DNA, and they invoke the right DNA based on the environment they're in. They can survive in nuclear waste. They actually like radiation environment. NASA uses them to test a lot of high radiation environments like reentry. So Dino is a really cool example of biological systems being able to adapt and change genes depending on environment they're in. And of course, systems biology. Biological systems are not simple targets because you're looking at a whole big system comprising of a bunch of little parts, and the whole system is intelligent. Each individual part is also intelligent. Everything talks with everything, and it evolves over time. So, not very simple. Another myth is if FDA approved it, that means everything, all the mechanisms are fully understood. We know what's going on. Kinda, but not really. The primary concern of Food and Drug Administration is safety. Secondary, but number 1.1, most important concern is efficacy. So safety and efficacy. Does the treatment hurt the target, the person? Does it kill them? Does it make things worse? It doesn't, okay. Does it work? Is there positive effect? Are there negative side effects? Does it cause cancer? So treatment or device can be approved without full and complete and comprehensive understanding of the actual pathway, the mechanism, the full mechanistic pathway by which the treatment works. So again, safety and efficacy are primary concerns. So when FDA approves something, that does not mean mechanism is understood. Furthermore, I think once FDA approves, especially de novo, like a new treatment, the scientists among us should really pay attention to those approvals and dive deep into the mechanisms. Hopefully, that research will prove that indeed this treatment is really amazing and has no side effects. And understanding that mechanism may lead to development of even better treatment. And anyway, regulatory pathways, they focus on outcomes and safety. Science is what's important to drive the mechanistic understanding beyond the FDA approval. Another myth is innovation, university innovation is purely scientific. Not really. Bioengineering, not only. Bioengineering is bioengineering. Engineering challenges, materials challenges, they're all highly important. More and more lately, I'm realizing how important the psychology of the whole thing is, as well, how the patients perceive the treatment. If the patient merely thinks that the treatment is working, the outcome of the treatment is usually frequently more positive if the patient believes in it. So human factors, they affect implementation quite a bit, and systems level thinking again is required. So it's not pure science. It is science. We all stand on shoulders of giants. The things that I'm able to do them because of major scientific breakthroughs in plasma physics, plasma chemistry, and plasma engineering. I'm able to take those amazing scientific achievements and start implementing them in bioengineering solutions. So what really matters? Dose rate, exposure control, mechanistic validation, reproducibility of your results. N equals 5. N equals 3 is minimum, which means number of repeats for each experiment. If you can do n equals 10, n equals 100, do that. And new discoveries in bioengineering, like an organ on a chip, allow us to do n of 1,000 without sacrificing 1,000 animals, which is absolutely freaking awesome. And anyway, bioengineering is a translational medicine discipline. So a lot of the work we do is to translate scientific discoveries to the bedside, to the patients, make human lives better. So in closing thoughts, bioengineering, as you might have guessed, is pretty complex and very interdisciplinary. Oversimplification often leads to errors, mistakes, and even bad patient outcomes. Understanding of mechanisms and measurement accuracy matter quite a bit. So, any myths you want me to examine next, I'm happy to answer your questions. Please post them down in the comments section. And if you have ideas for new videos, you have some questions you want me to dive into, please go ahead and ask them. Peace. I'll see you in the next one.